Hello, cell biology fans. Let's talk about glycolysis as we learned in class. Remember, glycolysis happens in the cytosol of a cell. So if I draw here the membrane of a cell, you need to have a way for glucose to be transported across that membrane. And the way that works is you might have a glut transporter. Remember gluts? Glut 1, 2, 3, and 4 transport glucose, and glut 5 is a fructose transporter. So what happens is you will have a glucose molecule, which I will draw here. It should be carbons in chemistry are denoted by black circles. Oh my goodness, I thought I got that correct that time. Oh, I see, I'm not doing it in the right place. Okay, let's try that. All right, so you have a 6-carbon molecule, glucose, that gets transported across the plasma membrane into the cytosol. And you end up with glucose in the cytosol of the cell. And what happens to that glucose molecule is that it gets converted into a 3-carbon molecules called pyruvate. The process whereby this 6-carbon molecule gets converted, right, through a number of steps into 2 3-carbon molecules is called glycolysis, and these 3-carbon molecules are called pyruvate. So what does this process look like? Let me make it slightly bigger, and over here on this side I'm going to draw for you the steps in glycolysis. The first step in glycolysis is the addition of a phosphate group onto the end of the glucose molecule. The process of adding phosphate requires the input of energy, and so ATP is used in this process to add the phosphate group to the 6-carbon molecule glucose, and that releases ADP. We would call this, it's an investment stage, because you are investing energy to ultimately, at the end of glycolysis, to generate molecules that can enter into the mitochondria to get converted into even more energy. The second step is very similar, and in this case what we're going to do is we're going to add another phosphate group to the other end of the molecule. Again, this added phosphate now, shown here, requires the input of energy in the form of ATP with the release of ADP. So at this point, you haven't converted any energy into ATP. You've actually used 2 ATP, so you're minus 2 ATPs. The molecule generated here is called fructose 1,6 bisphosphate, denoting the 1 and the 6 are the positions of the phosphate. Fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate gets cleaved into 2 molecules, each with 3 carbons. Each of the 3-carbon molecules has 1 phosphate group attached, based on the 6-carbon molecule with 2 phosphates that it's being split from. The next thing that happens is we actually are going to add another phosphate group to these 3-carbon molecules. And this is done with a molecule called NAD+. Let me get this smaller. So NAD, sorry, that should be in black. NAD plus generates some NADH. NADH is an electron carrier. And now you're going to see I'm going to draw this on both sides because we have 2 molecules in each step. The next things that happen are going to lead to the removal of the phosphate groups. And with the removal of the phosphate groups, we actually will convert the bond energies into the bond energy found in ATP. So if I push this up a little bit, we're going to remove a phosphate. So in this next step, nope, I keep drawing in red when I mean to be drawing in black. Alright, so that first step, you get some NADH. In the next step, we're going to remove a phosphate, and that is going to allow us to harvest the energy in a molecule of ATP on each side. That's ADP is going to go to ATP, right, with the phosphate that comes off, plus inorganic phosphate, plus inorganic phosphate. And then the next steps are to move the remaining phosphate into a position where it can be removed. And this is called an isomerization. Going from this 3-carbon molecule here with 1 phosphate to this 3-carbon molecule with 1 phosphate. And then the last and final step is the removal of that additional phosphate group. As you can see here, I've added in, once we remove that last phosphate group, we have another event where ADP is converted into ATP. So if I put boxes around all of the molecules that get generated, we have ATP, ATP, NADH. And so we invested in the first half of the glycolysis reactions, but in the second half we got the payoff. And the payoff is a little bit of ATP, so if you 4 ATPs here, that's 4 plus. But up above, we lost 2 ATPs. So the net product equals 2 ATPs. We've also generated some NADH, 2 NADH. And at the very end, we have the 2 3-carbon molecules, 2 pyruvates. And that's glycolysis. Pretty darn simple.